So process of rDNA technology, process of recombinant DNA technology. So in the previous section, so we had talked about the tools of rDNA technology. Now let us talk about the process of rDNA technology. So here we have different steps in the process of rDNA technology. It is very important for IPA as well as NEET point of view. So in this year 2025, so many questions from this chapter, biotechnology principles and processes, a very important chapter. So here these are the some of the important steps of process of rDNA technology. The first step is the isolation of DNA in pure form and fragmentation of DNA by restriction endonuclease. And third step is the isolation of desired DNA fragments by gel electrophoresis. And another step is the ligation of DNA fragment with a vector by DNA ligase. And another important step is transferring the rDNA into the host and culturing the host cells in a medium at large scale in a bioreactor and extraction of desired product by downstream processing. So these are the some steps of rDNA technology. Okay. So here restriction endonucleases are used to cut the desired DNA, gene of interest, or we can call it as foreign DNA or alien DNA. This is the gene of interest. For example, ఇన్సులిన్. సో హ్యూమన్ ఇన్సులిన్ తీసుకోండి. హ్యూమన్ ఇన్సులిన్ అనేది మన జీన్ ఆఫ్ ఇంట్రెస్ట్ అనుకుంటే, సో దట్ మే బి కాల్డ్ ఎస్ ఫారిన్ డిఎన్ఏ ఆర్ టార్గెట్ డిఎన్ఏ ఆర్ సోర్స్ డిఎన్ఏ. అండ్ హియర్ అగైన్ ఇన్ ఆర్డర్ టు క్రియేట్ ఆ డిఎన్ఏ వెక్టార్ ఈజ్ నెససరీ. సో వెక్టార్ అనేది ఖచ్చితంగా అవసరం ఉంటుంది. హియర్ ఇట్ ఈస్ అ జీన్ ఆఫ్ ఇంట్రెస్ట్, దట్ ఈస్ ఫారిన్ డిఎన్ఏ. సో హియర్ foreign DNA as well as vector DNA cut with same restriction enzyme, that is EcoR1. What is EcoR1? It is a molecular scissor restriction endonuclease which cuts the DNA at specific sequence. The sequence is known as the GAATTC, right? So here using the restriction enzyme, that is same restriction enzyme cutting both foreign DNA and vector DNA at specific point. So in last class we had discussed that exonucleases cut the DNA at ends, but endonucleases cut the DNA at specific locations. They recognize a specific sequence and cut the DNA. సో ఇక్కడ రెస్ట్రిక్షన్ ఎండోన్యూక్లియస్ అనేవి డిఎన్ఏలో స్పెసిఫిక్ లొకేషన్ గుర్తించి అక్కడ సీక్వెన్స్ని కట్ చేస్తారు అనమాట. సో ఆ సీక్వెన్స్ ఏంటంటే జిఏఏ టీటీసీ సీక్వెన్స్. సో ఇక్కడ ఫారెన్ డిఎన్ఏకి ఏదైతే రెస్ట్రిక్షన్ ఎంజైమ్ యూజ్ చేస్తున్నామో సేమ్ రెస్ట్రిక్షన్ ఎండోన్యూక్లియస్ ఈజ్ యూజ్డ్ ఫర్ ది వెక్టార్ డిఎన్ఏ. ఓకే. సో సేమ్ రెస్ట్రిక్షన్ ఎంజైమ్ cutting both foreign DNA and vector DNA at specific point, so that we obtain some fragments, DNA fragments, that is gene of interest as well as vector DNA. And here another enzyme, that is glue enzyme, molecular glue, usually called as DNA ligase. So this DNA ligase join foreign DNA to plasmid, that is nothing but vector or cloning vehicle. సో ఈ జీన్ ఆఫ్ ఇంట్రెస్ట్ వెక్టార్ డిఎన్ఏకి లైగేట్ చేస్తున్నాం యూజింగ్ ద ఎంజైమ్ డిఎన్ఏ లైగేజ్ which act as a molecular glue, so that we obtain our rDNA అనేది ఫామ్ అయింది. This rDNA is also called as a chimeric DNA or hybrid DNA or new DNA. Chimeric DNA or hybrid DNA or new DNA. So here this rDNA is must be transformed into host. ఈ rDNA అనేది హోస్ట్లోకి ట్రాన్స్ఫార్మ్ చేయాలి. సో ట్రాన్స్ఫార్మ్ చేయాలి అంటే ఇక్కడ మనకు డిఫరెంట్ మెథడ్స్ ఉంటాయి. దట్ ఈస్ హీట్ షాక్ మెథడ్. వన్ ఈజ్ అ heat and cold shock, and another method is a micro injection. Another one is a biolistic method. It is also called as gene gun method or biolistic method. Okay, so here heat and cold shock method. That means this rDNA and host incubated, and they are subjected to heat as well as cold immediately. So we have to repeat the experiment 7 to 8 times. So heat shock, again cold, heat, cold, so that rDNA gets transformed into this host. So this host with rDNA, and this host is cultured in a bioreactor, bioreactor to obtain the large quantities of our gene of interest, that is insulin. Okay, so here it is a diagrammatic representation of the rDNA technology, and heat and cold shock is one method. In another method, that is micro injection. So this rDNA is directly injected into the nucleus of animal cell. సో ఆర్డిఎన్ఏ అనేది ఎనిమల్ యొక్క న్యూక్లియస్లోకి డైరెక్ట్గా ఇంజెక్ట్ చేయబడుతుంది. అది మైక్రో ఇంజెక్షన్ మెథడ్. అండ్ అనదర్ మెథడ్ ఈజ్ అ gene gun method or biolistic method. In the gene gun method or biolistic method, here we use tungsten or gold particles. So this rDNA as well as host coated with the tungsten particles or gold particles, so that particle bombardment takes place between the rDNA and host, so that the rDNA is transformed into the host, and it is very expensive method. Right? So these are some of the important methods. Other than this, we have another method is also there, that is disarmed pathogen. Here, for example, Agrobacterium tumefaciens is a pathogen. Agrobacterium tumefaciens is a pathogen which can cause the disease in pear and as well as apple. The disease name is a crown gall disease, and this bacteria possesses plasmid. That name of the plasmid is TI plasmid, tumor induced plasmid. And here this plasmid is extracted, and here that is our gene of interest is inserted in this plasmid, and again reinsert into the agrobacterium, and then this is infected to plants, so that our rDNA is transformed into the required organism. And as well as in animals, we use retroviruses as a disarmed pathogens. Retroviruses for animals and agrobacterium for plants. Okay, so this is a fourth method, right? So first method is a heat and cold shock, and second method is a micro injection, third method is a gene gun method, and fourth method is that is a disarmed pathogen, so that rDNA successfully transformed into the host organism. Okay, right. So here then come to the steps. Right. So first step is a isolation of DNA. So here, so bacterial cell wall digested by lysozyme. So the DNA must be in pure form. So here bacterial cell wall digested by the enzyme lysozyme, and plant cell wall digested by the cellulase and pectinase, and fungal cell wall is digested by the chitinase, and RNA of the cellular content is digested by the ribonuclease, and proteins removed by the proteases, and purified DNA ultimately precipitated out after addition of chilled ethanol. The precipitated DNA is separated and removed by spooling. right? So here the DNA is in order to cut the DNA is available in the pure form. So in order to isolate the DNA, so cell membrane or cell wall must be treated with these enzymes. So not only these enzymes, some detergents also used to treat the cell wall or cell membrane, that is sodium dodecyl sulfate or some other detergents, or calcium divalent ions also used to treat the cell wall to separate or to isolate the DNA. So here if you take bacteria, in order to extract the bacterial DNA, the cell wall must be digested by the lysozyme enzyme. So like that, if you take plant cell, cellulase and pectinase can be used, and for the fungal cell wall, it is digested by the chitinase, because the component present in fungi is the chitin. And here in the similar way, the DNA must be needed in pure form. DNA అనేది pure form లో ఉండాలి కాబట్టి we should remove the other molecules like RNA, proteins, lipids, etc. RNA removed by the ribonuclease, RNAs. Proteins are removed by the proteases. Like that, so different enzymes are used to remove the different molecules. So because we require only DNA, and the DNA is, so here the purified DNA ultimately precipitated out after the addition of chilled ethanol. After the addition of chilled ethanol, so when we add the ethanol, the DNA gets precipitated. The DNA gets precipitated. It is available in a fine thread forms. Okay, so chilled ethanol. After addition of chilled ethanol, DNA precipitates ultimately, and this can be removed by using a glass rod. This can be removed by using a glass rod, and this process is known as the spooling. Very important, spooling. So the precipitated DNA is separated and removed by spooling. Okay, so these are very important for NEET point of view. So purified DNA ultimately precipitated out after addition of chilled ethanol, and the precipitated DNA is separated and removed by spooling. Right. Next, second step is a fragmentation of DNA by restriction endonuclease. Fragmentation of DNA by restriction endonuclease. So here this fragmentation of DNA by restriction endonuclease is carried by the restriction endonuclease, that is eco eco R1. So eco R1 is extracted from Escherichia coli. Okay, right. So here action of restriction enzymes. So the enzyme eco R1 cuts both DNA strands at the same time. Both DNA strands means that is vector DNA as well as foreign DNA. If you look at into the image, so here it is a vector DNA. It have a specific sequence, that is GAA TTC. One is 5 prime to 3 prime direction. One is 5 prime to 3 prime. Another one is 3 prime to 5 prime. It is the anti-parallel nature of DNA. So the sequence, if you read in a forward and backward direction, it reads the same. That is a palindromic sequence. Okay. So here it makes a cut between G and A, G and A, G and A. Okay. In the same way, in the similar fashion, here in the foreign DNA, that is gene of interest, it makes a cut between G and A in one strand, in another strand G and A. Right. So here eco R1 cuts the DNA between bases G and A only when the sequence GAA TTC is present in the DNA. That is eco R1 recognizes a specific sequence. The sequence is known as GAA TTC. So for every restriction enzyme, there is a particular recognition sequence. So for eco R1, it is a GAA TTC. For other enzymes, so there is another sequence. So here after cutting the DNA, it makes a sticky ends or cohesive ends. Sticky ends. So these are the sticky ends. So these sticky ends are joined using the enzyme ligase, DNA ligase. So these sticky ends are joined using the enzyme DNA ligase. So DNA fragments join at sticky ends. So that we obtain our recombinant DNA molecule or DNA molecule, right? So here the important thing is a separation. Next step is a separation and isolation of DNA fragments. సో కటింగ్ అనేది రెస్ట్రిక్షన్ న్యూక్లియస్ ద్వారా చేసినాము. ఇప్పుడు సపరేషన్ చేయాలన్నమాట. సపరేషన్ అండ్ ఐసోలేషన్ ఆఫ్ డిఎన్ఏ ఫ్రాగ్మెంట్స్ అండ్ దట్ కెన్ బి క్యారీడ్ ఇన్ ఏ అగరోస్ జెల్ ఎలక్ట్రోఫోరోసిస్. అగరోస్ జెల్ electrophoresis. So here it is a diagram, a typical agarose gel electrophoresis showing migration of undigested and digested set of DNA fragments lane 2 to 4. So here the cutting of DNA by restriction endonucleases results in the fragments of DNA, and these fragments are separated by a technique called agarose gel electrophoresis. So agarose is extracted from the red algae, Gelidium and Gracilaria. So here, since the DNA fragments are negatively charged, they can be separated by forcing them to move towards anode under the electric field through medium matrix, so which provide the sieving effect. The DNA fragments separate according to their size through sieving effect provided by agarose gel. Hence, the smaller the fragment size, further it moves. సో సీవింగ్ ఎఫెక్ట్ అంటే ఏం లేదు, ఈ అగరోస్ జెల్ అనేది సెమీ సాలిడ్గా ఉంటుంది. సో ఇట్ పొసెస్ పోర్స్. త్రూ దట్ పోర్స్ the DNA fragments can be moved easily. Okay, smaller fragments moved very easily and very fastly. So here DNA most commonly used matrix is agarose, which is a natural polymer extracted from seaweed. It is a previous NEET question. So most commonly used matrix is agarose, a natural polymer extracted from seaweed. That seaweed is nothing but red algae, Gelidium and Gracilaria. So DNA fragments separate according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, farther it moves. So if you look at into the diagram, you can clearly observe the fragments, right? Right. So here these are the wells. Our samples are loaded in these wells. So while loading the sample, ethidium bromide is a staining solution. It is also loaded along with our DNA fragments in order to visualize the fragments under the UV light. So here this is a larger fragment. It is a larger fragment. So usually larger fragments are present near the wells. And here these smaller fragments move very fast. And they are forcing towards a anode, because here this DNA is negatively charged molecule, and they can be separated by forcing them to move towards anode under an electric field through a medium or matrix, that is nothing but agarose gel. Okay, so here the separated fragments are visualized, very important. The separated fragments are visualized by staining them with ethidium bromide followed by exposure to UV radiation or UV light. Okay, so here ethidium bromide is a staining solution. So using that staining solution, when this agarose gel is exposed to UV light, one can easily observe the bright orange color bands. So the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece, and this step is known as the elution. Previous NEET question. So here the cutting of that DNA fragment from the agarose gel is known as elution. We can call it as elution. So very important this step is that you need to remember the staining solution as well as what is elution. Okay, so ethidium bromide is carcinogenic. While performing an experiment, you need to be very careful because it is a carcinogenic agent, right? So here this is about the separation of fragments using agarosel electrophoresis. Okay, next step is fourth step is here, right? Ligation of DNA fragments with a vector by DNA ligase. So vector DNA as well as foreign DNA ligated with the enzyme DNA ligase. And another important step is that amplification of gene of interest using PCR, very important. So gene of interest or source DNA or foreign DNA or target DNA. So here this PCR is act like a Xerox machine to produce billion copies of gene of interest, and it is 2025 NEET question. So examiner asked very logically. Okay. So now let us see amplification of gene of interest using PCR. So PCR stands for polymerase chain reaction. PCR stands for polymerase chain reaction, and it was discovered by the Carey Mullis in 1980s. And multiple copy of gene of interest can be synthesized in vitro. In vitro means in laboratory condition. So PCR includes following steps: denaturation, annealing, extension. Denaturation, annealing, extension. So here, right, so denaturation, annealing, extension. These are the three steps in PCR machine. So first, denaturation. So denaturation is nothing but so when this gene of interest is exposed to high temperature, that is double stranded DNA made single stranded. If you expose the double stranded DNA to heat, that is heating at 94 degrees centigrade, each single stranded DNA is called template strand. So this is a double stranded DNA or gene of interest. So this gene of interest is exposed to uh, heat or temperature at 94 degrees centigrade. The strands get separated. The DNA strands get separated, and each strand act as a template. Each strand act as a template. Okay, each strand act as a template. Okay, so here, so in the DNA replication, primers, RNA primase will add a small stretch of nucleotide that is called as primer. Here in the PCR machine, so oligonucleotides will act as primers. Oligonucleotides, so that is annealing steps. So two steps of primers, two sets of primer, that is small oligonucleotide chain that are complementary to the DNA at 3' end of the DNA template added to the medium. This is done at around 50 degrees temperature. 50 degrees temperature. So at two sides, 5' to 3' direction, and another one is 3' to 5' direction, the primers will be added to these strands, and next step is the extension. So extension is done by the tag DNA polymerase. Extension is done by the tag DNA polymerase. That is just like in the molecular biology, DNA replication process. So where the DNA polymerase is required for the extension of DNA strand. సో న్యూలీ సింథసైజ్ డిఎన్ఏ స్టాండ్ ఎక్స్టెన్షన్ కొరకు డిఎన్ఏ పాలిమరేజ్ అనేది అవసరం ఉంటుంది రెప్లికేషన్లో. సో ఇన్ ద సేమ్ వే హియర్ దట్ ట్యాక్ డిఎన్ఏ పాలిమరేజ్, ట్యాక్ డిఎన్ఏ polymerase, that is Thermus aquaticus. So this tag DNA polymerase is extracted from the Thermus aquaticus. It can tolerate the high temperature. So it is involved in the synthesis of new strand, that is extension. So here After extension, we get two cycles. After extension we get two strands, and like that the cycle repeats. So here the question asked by the examiner is, um, ఏ ప్యాటర్న్లో మనకు బిలియన్ కాపీస్ ప్రొడ్యూస్ అయితే అని అడిగిండు. ఏ ప్యాటర్న్లో బిలియన్ కాపీస్ ప్రొడ్యూస్ అయితే అని అడిగిండు. సో ఇక్కడ మనం తీసుకుంటే జీన్ ఆఫ్ ఇంట్రెస్ట్ అనేది వన్ జీన్ ఆఫ్ ఇంట్రెస్ట్ ఈస్ ప్రజెంట్. ఈ వన్ జీన్ ఆఫ్ ఇంట్రెస్ట్ నుంచి మనకు టూ జీన్ ఆఫ్ ఇంట్రెస్ట్లు ఫామ్ అవుతున్నాయి. టూ జీన్ ఆఫ్ ఇంట్రెస్ట్. సో దిస్ ఈస్ ద రైట్. సో దిస్ ఈస్ ద one gene of interest. So assume that 2 power 0 1. After completion of one cycle we get two strands, that is, so 2 power 1 is equal to 2 strands, 2 power 2 is equal to 4, 2 power 3 is equal to 8. So it follows pattern. The pattern is the 2 power n pattern. సో ఈ ప్యాటర్న్లో అడిగాను. అంటే ఎట్లా ఇచ్చిందంటే క్వశ్చన్, ఏ ప్యాటర్న్లో మనకు బిలియన్ కాపీస్ ఆఫ్ జీన్ ఆఫ్ ఇంట్రెస్ట్ ప్రొడ్యూస్ అవుతాయని. సో ఇట్లా N2, 2N అండ్ 2N. సో దిస్ విల్ బి ద రైట్ ఆన్సర్. సో సింపుల్ క్వశ్చన్, జస్ట్ లైక్ మైటాసిస్. సో మైటాసిస్లో మనం డిస్కస్ చేసినాం. సో వన్ సెల్ divides, produces two cells, two cells divides, produce four cells, like that it also follows the same fashion. So two strands form అయినాక again the cycle repeats. అట్లా మనకి ఎన్ని కావాలనుకుంటే అన్ని cycles ప్రొడ్యూస్ చేయొచ్చు. after so many cycles amplified amplified DNA is formed, that is 1 billion times or 1 billion copies can be produced using the PCR machine, just like PCR. Okay. సో ఈ మెషిన్ వల్ల యూజ్ ఏంటంటే, రీకాంబినెంట్ డిఎన్ఏ టెక్నాలజీ ప్రాసెస్లో మనం జీన్ ఆఫ్ ఇంట్రెస్ట్ యూజ్ చేసినప్పుడు ఒకవేళ ఆ డిఎన్ఏ ప్రాసెస్ ఫెయిల్ అయితే మళ్ళీ ఆర్గానిజంని బ్రేక్ చేసి డిఎన్ఏని ఎక్స్ట్రాక్ట్ చేయడము, అండ్ ఈ ప్రాసెస్ అంతా కూడా కొంచెం కాంప్లికేటెడ్ అనమాట. సో ఒకవేళ ఆర్డిఎన్ఏ ప్రాసెస్ ఫెయిల్ అయినప్పటికీ కూడా మనం మన జీన్ ఆఫ్ ఇంట్రెస్ట్ని తీసుకోవడానికి ఈ మెషిన్, పీసీఆర్ మెషిన్ అనేది యూజ్ అవుతుంది అనమాట. సో it is considered as the heart of the molecular biology, heart of the biotechnology. So this machine was discovered by the Carey Mullis. Even in corona time, this PCR machine is used to detect the genetic material, that is pathogen genetic material. Okay. సిమ్టమ్స్ అపియర్ అవ్వకముందే మన బాడీలో ప్యాథోజన్ ఉన్నదా లేదా అని చెప్పేసి ఈ పీసీఆర్ యూజ్ చేసి చెప్పొచ్చు అనమాట. సో దట్ మనం ప్రాపర్ ట్రీట్మెంట్ తీసుకుంటే treatment, so that we can get soon overcome the virus. Okay. Next step is the right. So these are the steps: denaturation, annealing, extension. And next step is the this transfer of rDNA into the host. So we had discussed already the transfer of rDNA into the host. That is, the methods are heat, cold shock method, microinjection, biolistic method or gene gun method, and another method is the disarmed pathogen. Right. So next step is the obtaining foreign gene product or recombinant product. So the protein encoding gene is expressed in heterogeneous host called recombinant protein. సో జీన్ ఆఫ్ ఇంట్రెస్ట్ ఏదైతే ఉన్నదో అది హోస్ట్లోకి ఇంట్రడ్యూస్ చేసినాక ద ప్రోటీన్ ఎన్కోడింగ్ జీన్ ఈజ్ ఎక్స్ప్రెస్డ్ ఇన్ heterogeneous host called recombinant protein, and the host is cultured in a continuous culture system providing in a bioreactor. సో బయోరియాక్టర్ అనేది ఒక లార్జ్ వెసల్. ఓకే. సో హియర్ we provide nutrient medium for the growth of the host, so that produces huge quantities of progenies, that is 1 become 2, 2 become 4, 4 become 8. So like that large quantities of our gene of interest can be produced in large quantities. So a bioreactor provides optimum growth conditions, that is temperature, pH, substrate, salts, vitamins, oxygen, etc. So this bioreactor convert the raw material into specific product and a specific enzyme. And next step is the downstream processing. Downstream processing. After biosynthesis, inside the bioreactor, the product has to be subjected through a series of process before it is ready for marketing. So the process includes separation and purification, which are collectively referred as a downstream processing. Very important. So downstream process includes mainly separation and purification. Separation and purification. The product has to be formulated in suitable preservatives. Such formulation has to undergo through clinical trials as in case of drugs. సో మనకు వచ్చిన ప్రోడక్ట్ని చిన్న చిన్న వైల్స్లో ఫిల్ చేసి క్వాలిటీ కంట్రోల్ డిపార్ట్మెంట్కి పంపిస్తే అక్కడ వాళ్ళు దాన్ని టెస్ట్ చేసుకొని మార్కెట్లోకి రిలీజ్ చేస్తారు అనమాట. ఓకే, రైట్. సో హియర్ నీట్ ఎగ్జామ్లో మనకు అడిగిన క్వశ్చన్, ఇంపార్టెంట్ క్వశ్చన్ ఏంటంటే, సో ఇక్కడ ఆర్డిఎన్ఏని హోస్ట్లోకి ట్రాన్స్ఫార్మ్ చేసినప్పుడు అది హోస్ట్లోకి ట్రాన్స్ఫర్మేషన్ జరిగిందా లేదా అని తెలుసుకోవడానికి ఏ మెథడ్స్ ఉంటాయని అడిగిండు. సెలెక్టబుల్ మార్కర్, దట్ ఈస్ యాంటీబయాటిక్ రెసిస్టెన్స్ జీన్స్, యాంటీబయాటిక్ రెసిస్టెన్స్ జీన్స్. సో హియర్ selectable marker. It is required to identify recombinant from the non-recombinant. It helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. So transformation is a procedure through which a piece of foreign DNA is introduced in host bacterium. Normally, the gene coding resistance to antibiotics such as ampicillin, tetracycline, chloramphenicol, or kanamycin, etc., are considered as useful selectable markers for E. coli. So the normal E. coli cell do not carry resistance against any of these antibiotics. Okay. So here, the recombinant can be identified from the non-recombinant in following steps. All are grown in ampicillin medium, and one replica of above plate grown in ampicillin medium, other replica grown in the medium containing both tetracycline and ampicillin. The colonies grows in plate 1 but fail to grow in plate 2 are identified as recombinants. So here, if we insert our gene of interest in the antibiotic resistance gene, for example, this is the plasmid. So it is a tetracycline resistance gene encoding region, and it is a ampicillin resistance encoding gene. So if we insert our gene of interest, to this region, that is tetracycline region, that is gene of interest, here this antibiotic loses its function. This tetracycline loses its function. Okay. So here the colonies grows in plate 1 but fail to grow in plate 2 are identified as recombinant. So here, if we grow this host in the ampicillin medium, it can survive. But if it grow in plate 2, that is tetracycline medium, it cannot. If the colonies grow in plate 1 but fail to grow in plate 2, that means that plate 2 contains a tetracycline medium. So ఈ tetracycline mediumలో grow చేసినప్పుడు it cannot survive. So that we can easily differentiate between transformants of recombinant and non-transformants of recombinants, okay, by using this plating method. And another method is insertional inactivation. ఇది కూడా 2025లో అడిగిన క్వశ్చన్. దట్ ఈస్ 2025 అడిగిన క్వశ్చన్ ఇన్సర్షనల్ ఇన్యాక్టివేషన్. సో దట్ ఈస్ ఆల్సో కాల్డ్ యాజ్ బ్లూ వైట్ స్క్రీనింగ్ మెథడ్. బ్లూ వైట్ screening method, right? So here in this method, if gene of interest is inserted near the lac Z gene, lac Z gene is responsible for the synthesis of beta galactosidase. This gene is responsible for the beta galactosidase, right? So if this bacteria or this host grow on X galactose medium మీద గ్రో చేసినప్పుడు బ్లూ కలర్ కాలనీస్ ఫామ్ అవుతాయి. బ్లూ కలర్ కాలనీస్ విల్ బి ఫామ్, రైట్? ఇప్పుడు జీన్ ఆఫ్ ఇంట్రెస్ట్ లేనప్పుడు, జీన్ ఆఫ్ ఇంట్రెస్ట్ అనేది ఇక్కడ జెడ్ జీన్ దగ్గర ఇన్సర్ట్ లేనప్పుడు this can produce the enzyme beta galactosidase, and if this grown in the medium which contains the X galactose, it produces the blue color colonies, right? It is a non-transient host, non-transient recombinant host, okay? Non-transient or non-recombinant host, it produces the blue color colonies if the gene of interest, if the gene of interest is present, or if we insert our gene of interest at the Z region, it cannot, the Z loses its function, just like tetracycline region లో gene of interest insert చేసినప్పుడు tetracycline ఎట్లయితే దాని యొక్క ఫంక్షన్ లాస్ అయిందో, ఇక్కడ కూడా this lac Z gene loses its function, and it cannot able to synthesize beta galactosidase, so that blue color colonies will not be formed. So here the colonies, white color colonies or colorless colonies will be formed. So white color colonies or colorless colonies will be formed. That is when the gene of interest is present. So it is another method, that is a blue white screening or insertional inactivation method. So ఇది మనకు రెండు క్వశ్చన్స్ అడిగింది 2025లో, వెరీ ఇంపార్టెంట్. మోస్ట్ ఆఫ్ ద స్టూడెంట్స్ స్ట్రగుల్ ఇన్ దిస్ ఏరియా, ఓకే, రైట్. And another method is present, that is radioactive probe to detect the transformed host, okay? So this method is not given in our NCERT, so no need to bother about this one. Just remember that one is a selectable marker antibody resistance gene, another one is a blue white screening method.